Lab 25, Mitosis in Plants. Introduction. In flowering plants, 3 types of structures are found above ground: leaves, stems, and flowers. The stems act as support for the plant. They also conduct water and carbohydrates to different parts of the plant. The leaves produce carbohydrates through photosynthesis. The flowers are used in reproduction. The below-ground parts are called roots. Roots are structures that usually penetrate the soil and spread downward. The root system absorbs water and nutrients. Also, the roots store food and anchor the above-ground parts to the plant. During the growing season, plants do not grow everywhere at the same time. Growth on the plant is confined to meristems. As you descend down the stem, the descendants of meristem cells are maturing or have already reached maturity. Growth of roots and stems occurs at the meristems because the new cells are being created in this region. The process of creating 2 cells from one for growth involves 2 processes: mitosis and cytokinesis. Mitosis is the duplication of the genetic material, and cytokinesis is the division of the cell membrane. The process of mitosis involves several steps. First, the strands of DNA in the nucleus must be duplicated. The DNA then coils into structures called chromosomes. Next, the nuclear membrane breaks down and the chromosomes move to the center of the cell. The chromatids on each chromosome separate. Each daughter cell receives an exact copy of the DNA from the parent cell. Cytokinesis involves the division of the cytoplasm and the separation of the newly formed daughter cells. Cell division for growth occurs in both plant and animal cells. However, there are differences. Most animals lose the capacity for continued growth upon reaching maturity. Even though new cells are formed to replace old or injured cells, repair and regeneration, skeletal growth is normally completed. This is called determinate growth, meaning that there is a point where cells lose the ability to increase in number. Most plants, however, demonstrate indeterminate growth. They continue to grow over their entire lifetime. The prepared slides used in this lab are allium root preparations. Allium is a member of the onion family. The roots of this plant are an area of rapid growth. The plant's roots are actively growing and moving through the soil in response to water and nutrient levels. More nutrients and water leads to more growth in the root cells. Fewer nutrients and water leads to slower growth. Procedure. Read the introduction to the lab before beginning any of the exercises. Handle prepared slides with care. Remember to use all of your lab drawing rules. Exercise 1. Mitosis in plants, onion root tips. 1. Obtain a prepared slide of the onion, Allium cepa. The slides are a cross-section through the root tips. Examine the slide with the microscope on low power. Locate the root cap, a thimble-like mass of cells at the tip of the root. Just above the root cap is an area of rapidly dividing cells called the apical meristem. See the illustration above. The cells in this region have a dense cytoplasm and large nucleus. Cells in this region will be square in shape. 4. Locate the cells in the apical meristem and switch to high power. 5. On high power, locate a cell that is not dividing. Its nucleus is in interphase. The nuclear membrane is clearly present, and the chromosomes are not evident. 6. Draw what you see in the field of view. Label the nucleus, cytoplasm, organelles, and the DNA. 7. Find cells that show the following mitotic phases. Title each drawing and label the chromosomes, or chromatids if the chromosomes have split. The cytoplasm, cell wall, and cell membrane. You won't be able to see it, but you know where it is. Prophase. The DNA has duplicated and the chromosomes are starting to form and become visible. The nuclear envelope is disappearing or is entirely absent. Metaphase. The duplicated chromosomes are aligned in the center of the cell. Anaphase. The chromatids separate at the centromere, are beginning to move to the edges of the cell. Telophase. The chromatids are at the edge of the cell, and the cytoplasm begins to close in and separate the parent cell into two daughter cells. Exercise 2. Quantity of cells in mitosis. In this exercise, data will be gathered to determine the number of cells in the apical meristems that are dividing at any one time. 1. On high power, count all the apical meristem cells you see. 2. Now count the number of cells that appear to be in some phase of mitosis. Record your results on the table in the handout. 3. Calculate the percentage of cells dividing by dividing the number of cells dividing by the total number of cells. Your answer should be a decimal less than 1.